Biology 9700, paper 21, May June 2024, question number 4 to question number 6 and part 2. Question number 4, figure 4.1 is a scanning electron micrograph showing the tissue that lines the bronchi in the gas exchange system. So 4.2 is a transmission electron micrograph of a horizontal section made at the position indicated by the two arrows. So two arrows has been shown over here. Name the cells labeled A and B. So two arrow shows the two parts which are labeled A and B. So we are going to write the name of the parts of the labeled A is ciliated epithelial cell. Ciliated epithelial cell. And second one is the goblet cell. So now we are moving towards the next part. Describe how the tissue shown in figure PowerPoint 1 is adapted to its function in the gas exchange system. And here you can see the ciliated epithelial cells. They will move. Epithelial cells will move mucus by their movement in the upward direction. And goblet cells are meant to produce mucus which traps dust particles and microbes and and stop them to enter, because ciliated epithelials are you can see hair-like structures which help the mucus to move in the upward direction into the throat. And secondly, the goblet cells, they are meant to produce the mucus. So the mucus is a sticky substance which is produced inside the respiratory tract. And this mucus is actually sticky to capture or to trap the dust particles and the other microorganisms which try to enter into the respiratory tract. The next question is, the structure labeled X have a characteristic internal appearance as seen in figure 4.2. 4.2, describe the internal appearance of the structure labeled X. Structure labeled X. You can see we are moving towards the picture. Structure labeled X. So you can see these. Now we are going on the space where we are going to write the answer. So the structure labeled X, internal appearance of the structure labeled. So it is, you can see 9 plus 2 pattern is seen over here. You can see over there. 9, 1, let me, 1, 2, 3, 4, 5, 6, 7, 8, 9. So these are the 9 doublets plus 2 which are in the center, they are single, plus 2 singlets. So 9 plus 2 pattern is there. It can be shown in picture. So you can see pattern is this. There are 9 doublets here, 9 doublets and 2 singlets in the center. So this appearance show 9 plus 2 pattern. This is called as 9 plus 2 pattern. So it is actually the arrangement of microtubules, which is present in the flagellum or cilia. So this is arrangement is present in flagellum and cilia. And next question, explain how figure 4.2 shows that each of the structure labeled X are intracellular. And you can see in around every 9 plus 2 pattern, there is a cell surface membrane. So they are bounded by cell surface membrane. So anything which has to be released, so we can say this is the arrangement which is called as intracellular. So it takes place inside the cell. So it is the intracellular arrangement because every cell is wrapped in cell surface membrane. Now the next question. Stem cells are found in the lining of the bronchi. Describe the function of the centrioles and explain how they are involved in the cell cycle of the stem cycle. So you can see the centrioles are only present in the animal cells, and when the cell cycle start in the prophase one, they move, they are replicated, they are paired, so they become 2 pairs and one pair, and each pair move towards the opposite poles. When they reach at the opposite poles, there is the ester formation, and they help in the formation of spindle fibers. So collectively they will make spindle fibers with the help of microtubules. So in this way they are helpful in the formation of spindle fibers. And then at the time when the chromosomes are get separated from mother, or they are divided at the time of cell division, when the centromere is divided in the anaphase and they move towards the opposite pole, at that time these microtubules or these centrioles are helpful in the movement of the chromosomes. So here I would like to say that centrioles organize organize microtubules and they each and they replicate during the cell division, and each pair move towards the opposite poles. And then what happens with them? In the movement of of chromosomes. So in this way, these centrioles are involved in the basic function at the time of cell division. So they are always available in the time of uh, cell division, and they are in the form of pairs. Now the next question, question number 5. And question number 5 is the pressure of water vapors inside and outside leaves can be measured. The difference between these pressures is known as the leaf vapor pressure deficit. Leaf vapor pressure deficit, LVPD. LVPD is one of the factors that influence the rate of transpiration. Scientists measure the effect of changing the LVPD on the rate of transpiration in several species of flowering plants that live in a variety of different habitats. Two of these species were Nerium oleander, a species that is adapted to grow in hot, dry conditions. On the other hand, Helianthus annus, a species that is not adapted for survival in hot, dry conditions. One species can survive in hot, dry conditions, the other cannot. So it shows the effect of increasing the LVPD. LVPD, you can see leaf vapor pressure deficit. So other factors were kept constant. So they have, there is a graph shown in the picture. You can see the dotted line shows the Nerium oleander and the straight line show the Helianthus annus. So you can see both of these curves, both on the, they starts from zero. Leaf vapor pressure deficit, LVP D is shown on x-axis, and whereas on y-axis is rate of transpiration. Now let's move towards the question regarding this. The question is, compare the results of the two species shown in figure 5.1. So in figure 5.1, you can see the differences. So the difference, first of all, you can see the difference is shown there is no transpiration when there is zero, and you can say it is increasing when when leaf vapor pressure LVPD is increasing. So in this way we can write that there will be no transpiration, when there is zero LVPD. And the second, you can say it is increasing in both of these curves, transpiration rate until 2.5. You can say 2.5. This is the, it increases until 2.5 LVPD. So at 0 LVPD, there is no transpiration. When the LVPD is increases, the rate of transpiration is also increases, which reaches as 10, which is maximum in both, maximum in case Helianthus annus, and the other it is low as it is 4 point or around 4.9 something. So it is in Nerium oleander. It means there is great difference in the range. And secondly, you can say the transpiration rate is higher in Helianthus annus as comparison to the other species, which is Nerium oleander. It means both of these species shows the variations in the LVPD, and due to LVPD, there is the difference in the rate of transpiration. So we are moving towards the next question, and next question is figure 5.2 shows part of the plant of Nereus oleander, and you can see this plant on screen, and shows a green section, cross section of a part of an oleander leaf. So you can see and they have marked 1 point. This is the some part of it mentioned with N. Now they have asked some questions regarding this. They have highlighted this part N, and you can see the difference in structure. So state and explain two adaptations shown by the leaves of an oleander that are visible in figure 5.3 and 5.4. So this is figure 5.3, and you can see this is the figure 5.4. So we have to write two adaptations. Two adaptations can be written as uh, 1, 2, 3, 1, 2, 3. So you can see these cells, they have thick the layers of the cells are thick layered. Thick layered cells. Uh, Thick cell wall. So why the cell wall is thick? Because it reduces the rate of diffusion of water vapors to atmosphere. Due to thick layers, it is adopted, that is they will be it reduces the rate of transpiration. And the second other, which is the other adapted function, the in figure 5.4, in 5.4 you can see the other is stomatas, no stomatas on upper surface or hidden stomatas. And what is their role when there are you can say low come stomatas, less stomatas, or uh, you can say no stomatas. So plant, it will protect itself from dehydration. And another point is the thick waxy cuticle. So in this way, dehydration by reducing diffusion. Now the question number next, question number 6 is there, and you can see the question number 6. Antibodies are produced by plasma cells. Figure 6.1 shows antigen bound to antigen binding sites of an antibody molecule. Hinge region is there, antigens and light chain, and these are the heavy chains. Two light chains and two heavy chains are there. You can say this is the structure of antibodies, and there is a place where the antigens attach themselves, and there is a hinge region in the center. So explain how the structure of an antigen binding site make it specific to a particular antigen shown in figure 6.1. So antigen binding site, you can say it has a complementary shape. So the antibodies and antigens have complementary shapes. which makes it specific to a particular antigen. Because the other which are not complementary to the shape, they will not attach to that site. Only the antigens which are complementary to this shape or which have the same shape, they will be attached to the that antigen binding site. Otherwise, they will not be attached. So the different substance or the different amino acids which are different in shape, they will not be attached. State the function of the hinge region of the antibody. It produces the flexibility for binding. It maintains flexibility. Now we are moving towards the next question. And next question is, the antibodies can bind to membrane receptors on cell of the immune system, such as macrophages, suggest an advantage of antibodies binding to receptors on macrophage. Okay, actually, antibodies binding to receptors on macrophage, because in this way, uh, it will be easy for the macrophages to engulf. So they are over here in this way. So destruction of the pathogens will take place. It will be very easy for them to destruct the or to kill the or to neutralize the pathogens. Easier for macrophages to engulf pathogens. Next question. It is estimated that the immune system of each person can make enough antibodies to bind to over 10 raised